Assalamualaikum and a very good morning. My name is Nurul Ainin a biomolecular science student. I made a study about the glycoprotein analysis of antibiotic-resistant Pseudomonas aeruginosa isolates from a curd. Pseudomonas aeruginosa is a gram-negative bacterium that have a root-shaped structure, lack of spore, and a single flagellum that can threaten the life of patients with cancer, severe burns, and cystic fibrosis. Pseudomonas aeruginosa produce protein which are necessary for biological processes and aid in resistance. The covalent and typically enzymatic modification of protein that occurs right after the production is known as protein post-translational modification or PTM. The most prominent type of PTM is glycosylation, which is the process of attaching monosaccharides to protein. Antibiotics have great chemotherapeutic potential and can cure a variety of microbial infections in both human and animal, since they work specifically against bacteria, which is in this study I use Pseudomonas aeruginosa. Therefore, for future management, the role of glycoprotein alteration in antibiotic-resistant isolate is very crucial. The first objective in this study is to characterize the antibiotic-resistant profile of local Pseudomonas aeruginosa isolates. Then, the second objective is to measure biofilm formation of Pseudomonas aeruginosa. For my third objective is to compare the glycoprotein profiles and correlate with antibiotic resistance. Firstly, to conduct the study, the media that need to be used was prepared, which were Miller-Hinton agar MH8 and Neutron agar NA. Both of these agar was used in the culture of bacteria and this division. Next, the Pseudomonas aeruginosa was cultured and grown by streaking it on NA plates, which then incubate overnight at 37 Celsius. Then, for this division, the bacteria was streaked on the MHA to determine whether it is resistant or susceptibility to the antibiotic given. In this study, I used aztreonam and ceftazidime. Next, the biofilm formation was conducted by using 96-well microtiter plate before checking the optical density OD by using thickened microplate reader. After that, protein extraction and separation was conducted by extract the total and cellular protein, Bradford protein quantification assay, acetone precipitation, and protein sample preparation. Then, to observe the band of protein, STS phage gel electrophoresis was run for 90 minutes at 200 volts. To view the glycoprotein band, the gel staining was run by using ProQ Emerald 300 glycoprotein gel stain. Next, we move on to the result in discussion. This division was used to observe whether Pseudomonas aeruginosa is resistant or not to the antibiotic they was using, which is in this study I used aztreonam and ceftazidime. Then, as a result, they shown only 3 samples of Pseudomonas aeruginosa were resistant to both antibiotic, as their zone of inhibition is nearly 0.0 mm. While for the other sample, 4 of them is partially susceptible and another 3 is susceptible. Biofilm formation was observed to determine the resistance of bacteria toward the antibiotic, and the greater the optical density OD was, the stronger the biofilm production is. Most of the sample are the moderate producer of biofilm, as it has greater OD than negative control, but have lower OD than positive control. Glycoprotein band can be observed, but it is not clear and have a little bit of smear. It might be because of the type of gel, which is 10% bis trace gel was not compatible with the glycoprotein stain, which resulted the band to smear at another well. Thus, the outer membrane protein cannot be observed, and the loaded sample also lower than it was supposed to load. But the protein band that might be observed in this study is lactin protein, which has molecular weight between 45 kilodalton to 57 kilodalton. This lactin protein in Pseudomonas aeruginosa might be helpful in treated patients with cystic fibrosis and respiratory tract infections. It can be concluded that the antibiotic-resistant profile of Pseudomonas aeruginosa isolates was characterized by conducting this division and the biofilm formation to observe whether the Pseudomonas aeruginosa resistant to selected antibiotic. However, the concentration or amount of sample have to be higher to make sure the zone of inhibition can be seen clearly. Nevertheless, sample 2, 8, and 10 were resistant to both antibiotic, and most of the sample are moderate producer of biofilm. Moreover, the glycosylation changes that may be related to antibiotic resistance occurred as protein leg B changed from 33 until 34 kilodalton to 42 until 57 kilodalton. But to obtain the result more clearly and accurate, the gel that was used must be compatible with the desired staining and ensure the power pack is in a good condition when run the gel electrophoresis. That is all from me. Thank you for listening to my presentation.